Good morning, or good afternoon, or good evening. Thank you for coming to the Polycultures and Pollinators online training. The purpose of this training is to make you a little bit more familiar with the study that we're conducting and to tell you a little bit more about native and wild bees and how they might promote pollination services in diversified cropping systems. My name is Elias Bloom, and my advisor is David Crowder. We developed this content and this project. We are both from Washington State University, and we are from the Department of Entomology. This project is part of a larger initiative called the Northwest Pollinator Initiative. We've been studying pollinators in small-scale farming systems for about the last 4 years. The project is funded by the Western Sustainable Agriculture Research and Education Program, Western SARE, the USDA, United States Department of Agriculture, and the National Science Foundation. We are also part of a collaboration with the Common Acre, which is a Seattle area non-for-profit. Once again, thank you for coming. This presentation will take about 20 to 30 minutes, and so we'll go ahead and get started with a little introduction on pollinators. And please excuse any delays between slides. There might be some short delays. Okay, so pollinator communities are responsible for most pollination on the planet. This means that no one species alone is responsible for the pollination services that these crops receive. In this figure, you can see that this author put together a number of different major agricultural crops throughout the world and looked at studies that surveyed the different pollinators that were responsible for pollination. And what this author found was that some crops, such as those represented by green, heavily depend on honeybees, so one species of pollinator, but many of these other crops throughout the world rely on wild insects, and at sometimes wild insects and honeybees, managed honeybees, to provide pollination. And this underscores the fact that wild pollinator communities are really quite important to pollination services. And what is a pollinator community? Well, a pollinator community are lots of different species, like these species over here on the right, working together to provide the pollination service to those crops. And so you can see that pollinators come in a variety of different shapes and sizes, and these are all wild bees that you can find right here in Washington State. So this isn't to scale, so these aren't all the same size like they appear here. Some of these bumblebees can be quite large, but you can see that they are incredibly diverse in their morphology. Their shapes and sizes are considerably different, their coloration is considerably different, and they all in some way or another help to provide pollination to plants. So this pollination is worth billions of dollars every year, and that's on an annual basis. And we know that there are thousands of bee species worldwide, probably somewhere around 4,000 bee species worldwide. And then in the Pacific Northwest, we have between 600 and 900 different species. Largely, the status of these wild bee communities in the Pacific Northwest is unknown. And the reason that is because much of the research on pollinators has happened within the last 100 years here in the United States. And a lot of it started in the eastern part of the United States and it slowly moved westward. So this indicates the need for increased monitoring, and that's part of the objectives for this project, is to increase the monitoring of native and wild pollinators to better our understanding of their community ecology and long-term trends in Washington State and the Pacific Northwest more broadly. A reason why these monitoring techniques are needed is because that wild bees are becoming increasingly rare or even extinct due to habitat loss. And so in North America and Europe, we've seen some large rain contractions, and we've also seen numerous extinction events. And that has led to the protection of some of these bee species, but in other places, there has been a significant loss of bee species, and that might be contributing to losses in pollination. These losses in bee abundance has been particularly strong in agricultural regions or farming regions, which has made pollination increasingly uncertain. So in these maps, this top map you can see in areas in yellow are the areas where bees are now predicted to be very, very low in their abundance. And then down here are areas that produce food that rely on pollinators, and areas that are in green are the areas that rely on pollinators, and that pollination service has become increasingly uncertain. So a lot of people want to know why these losses have occurred. A lot of it is due to the loss of habitat, and that loss of habitat has come because of the large scale conventionalization of farming systems. So these large farming systems have destroyed natural habitat for bees. And then within these farming systems, there's tremendously low crop diversity. And crop diversity is probably quite important to bee biodiversity, especially flowering crop diversity, because each of those different flower species provides a pollen resource for different species of bees. And that's something I'm going to get into a little bit more on the next slide. As I mentioned in the previous slide, crop diversity promotes wild bee biodiversity, and this is a huge result from our initial 4 years of studying pollinators, in that we found that with increasing plant or crop diversity on farms, seen with this black bar here, high plant diversity, you also significantly increased, these farmers also significantly increased bee diversity or bee genera on their farms. And this was over farming systems that had low biodiversity, low plant biodiversity. Those had significantly fewer bee genera on their farms. And so it's fairly well established that plant diversity correlates strongly with bee biodiversity, but this hasn't been shown very much in farming systems, so this is kind of an interesting result. And the reason that this happens is because plants provide unique niche that provide or facilitate for this interaction. So each plant species that has a flower is a unique niche or a niche for a pollinator, and sometimes only one species of bee will feed on one species of plant, and as you increase those plant species, you get increasing bee biodiversity because it provides resources for those unique interactions. However, it has remained to be unresolved as to whether or not increased plant diversity and bee biodiversity also increases pollination. And so that's the premise of this study, is that we want to figure out whether having increased plant biodiversity also continues to increase bee biodiversity, and whether or not that increasing bee biodiversity also increases pollination in these farming systems. And that's the polycultures and pollinators. The premise of polycultures and pollinators is to figure out whether or not increasing plant diversity also increases pollination services in diversified farming systems. And we suspect it will, but there are a number of ecological theory that underscore the relationship between plants and pollinators that would indicate that this would be the case. However, with losses in bee biodiversity it may be that pollination services in diversified farming systems become more limited. And so we want to see what's going on and kind of monitor these trends so we can begin to address any conservation and restoration needs for farming systems that might need those additional bee species to provide pollination to their plants. We've been working with citizen scientists to help collect valuable data to already unravel some of these complex interactions between plants and pollinators. So much of our citizen science work has happened in the Seattle area. And in the right map, you can see these different citizen science research sites that have been taking photographs of the interactions between plants and pollinators in their urban gardens. And at this one site, you can see in this upper left-hand corner all of the different bee groups that this citizen scientist has recorded with photography. And in addition to that, the citizen scientist has also recorded the different plant species these bees are interacting with. So we've been monitoring bees using photography, mainly in the Seattle area. And then also in the Seattle area, we've been working with citizen scientists to use artificial nests to monitor the habitat needs of cavity nesting bees like mason bees. And so in the bottom part on this right-hand side here, you can see some of the cavity nests that citizen scientists have put up. And then also on the left-hand side, you can see some of these data that the citizen scientists have collected. So now that we've worked in these different types of citizen scientist areas with photography and also with these artificial nests, we're now moving into the citizen science project with Polycultures and Pollinators that's going to be using non-destructive sampling techniques to monitor bees in a very quantitative kind of way. And in addition to that, we're looking to use sentinel plants to measure not only the bee biodiversity with the sampling, but also the pollination that's occurring in these farming systems. On the next slide, I'll be beginning to tell you a little bit about the methods for this project. And so it's important that you open up the methods document and follow along there, as the methods document has a little bit more detail to it than the PowerPoint presentation. I will certainly try to help to fill in some of the gaps that might be missing in both of the documents and answer any questions that you might have up front. But if you have any questions after the PowerPoint presentation and after reviewing your methods document, please let me know and via email, and I'll be happy to assist you in figuring out any of the methods that might not be very clear in either the PowerPoint presentation or in the handout. So the first method that I am asking you to do is to establish your sentinel plants to collect your fruit set data later on in the season. So you will be given the Dark Star zucchini seedlings, a 15 milliliter vial of fish emulsion, a slow release fertilizer, and red stake flags. All of this will be coming to you via mail. So the methods for this are to transplant your seedlings and work in the slow release fertilizer in the surrounding area. I'm trying to standardize the soil environment to reduce differences in productivity due to differences in soil productivity. You can space the plants 3 to 4 feet apart as you normally would with zucchini, and then you'll want to flag them so they don't get lost over time. I've visualized in my development of this project that you would plant these plants at the end of a row or somewhere in a row of zucchini or other squash plants, but you can put these essentially anywhere in your farming system or in your garden, wherever it works best for you. After you've transplanted your seedlings, which are in peat pots, so you just transplant them straight into the ground, no need to remove the peat pot, you will fertilize with the 15 milliliters of fish emulsion. and that's enough fish emulsion for 2 gallons, and it should be enough to water in these seedlings. After you've watered in your sentinel plants, go ahead and water and harvest your fruit ad libitum, so as per normal. And you can continue to do that until Monday, July 31st, 2017. And the reason for this is that these are just essentially becoming established plants in your farm, and we're going to give them about a month and a half to become fully established before we start the experiment. And so Monday, July 31st, 2017 is the start of the experiment. So on that date, I want you to remove all fruit regardless of size, but you can leave all flowers that are present on that date. And so all flowers that set in the next 2 weeks after July 31st, all flowers and fruit can be left on the plant, and that fruit over the next 2 weeks is how we're going to measure differences in pollination services across all of the farming systems that are involved in this project. So also on July 31st, if temperatures are above 55 degrees Fahrenheit, you're going to proceed with the sampling of your bee community. Now, if temperatures are not above 55 degrees Fahrenheit, or other weather conditions are not good for bee sampling, some other negative weather conditions are sustained wind speeds above 10 miles an hour, or cloudiness, we will be putting off sampling till the next most suitable day. And I will be emailing you ahead of time as a reminder to do the sampling and also to remove all the fruit on your sentinel plant. So I'll be keeping an eye on the weather, and if weather isn't looking good, then we can delay sampling until the next best day. Okay, so to sample the bee community, you're going to be using some traps that I will be mailing you. And the traps are going to measure the bee community that are contributing pollination services to your sentinel plants on that day. And so provided in your box of supplies, you'll be getting a water bottle that I've gotten from our recycle bin here in Puyallup. I will also be providing for you Blue Dawn dish soap, bee bowls, which are these small fluorescent bowls that are used to trap bees that are flying close to the ground, a blue vein trap and string, a bamboo tripod, and then also you'll be needing your data sheet, which can be found at the back of, actually it's going to be attached as an additional document. So I will be providing you with your data sheet too. So on July 31st, or your next suitable day, you're going to set up these traps from 8 a.m. till 5 p.m. Like you can see in this picture over here on the right hand side, I envision that you'll position your tripod over the sentinel plants. And so imagine that there are large zucchini plants in this area here, and that this tripod is directly located over your sentinel plants. And so from this tripod, you'll be hanging your blue vein trap, which is this trap here, and you'll be doing that with the piece of string that I've provided. So what you can do is thread your string through the vein on the blue vein trap, these plastic wings that are associated with the blue vein trap. You'll have to do a little bit of construction of your blue vein traps, and then you can tie it over the top of the tripod. And it's important that when you do this, you don't cover up the foliage, cover up the blue vein trap with the foliage of the plants, and that it's visible above your sentinel plants. Don't put it down in the foliage because the bees won't find it down there. It needs to be visible once you set it up. So the next thing you'll do will be to set out your bee bowls alongside your sentinel plants, and I envision that if the tripod is set up in the row, then the bee bowls are set up in the alley alongside of the sentinel plants. And again, make sure that the bee bowls are visible, as they need to be visible for bees to see them and then to get trapped in them. The next thing you'll need to do is mix 1 to 2 drops of your Blue Dawn dish soap in your plastic water bottle and add water. And then you'll essentially make a soapy solution there. You need to shake that up, and then you can just pour that soapy solution into your traps. And you'll need to make the soapy solution a few times, probably once for your bee bowls, and then you'll need to make another soapy solution for your blue vein trap. With your bee bowls, you only need to fill up the bee bowl to about 1/4 of an inch above the rim, but don't underfill them, especially on warm days. They can evaporate, and then bee species will be lost because the traps won't be able to capture them. And then in the blue vein trap, you only need about 1 inch of water in the bottom of the blue vein trap. And then on your data sheet, you'll want to note the date that you're doing the trapping, as well as the number of flowering crop species in your garden. And you can just estimate this like you did when you signed up for the project, but I just want to know how many flowering crops you have going on your farm or in your garden at that time, so you can just make an estimate of the number of flowering crop species. After that 8 to 5 p.m. period, imagine it's the end of your day, you're going to be collecting your bee samples for identification and to correlate with the fruit set data that you're also going to be collecting. So with that water bottle, you can cut the top off of it to make a funnel. And then I've also given you coffee filters and labels and also leak-proof containers. I want you to note that here it says I've provided you with 40 milliliters of 70% ethanol, and I'm not allowed to mail you ethanol without a special, it's like a special permit. So instead of using ethanol, you can go to any grocery store and buy Everclear, and Everclear is the best substitute for ethanol. And so I would just go to the grocery store and buy yourself a small amount of Everclear before you do these methods. So when you're ready to collect your specimens, cut off the top of the water bottle to make a funnel, and then you can place one of your coffee filters into the funnel and then pour all of your bee bowls into that filter. And like I said, I've given you a label that says bee bowls on it. Drop that label into that filter. And then after you've done that, after you poured all of the bee bowls into that filter, you can remove that filter and fold it up and place it in your leak-proof container. Then take your second filter and place it into your funnel, and then you can pour your blue vein trap into that filter. And once the blue vein trap is empty, you'll add the blue vein label to that filter. And throughout this process, if you're doing this into your sink, then you won't need to empty the reservoir that you've created when you've cut off the top of that water bottle. I call that bottom part the reservoir. But if you are emptying your traps through the funnel and into the reservoir at the bottom of that water bottle, then you'll need to empty that reservoir a number of times as you empty your traps. So once you've placed your filters into the leak-proof containers, you're going to pour your ethanol, but in your case, the Everclear that you're going to buy from the store, into those leak-proof containers and cap them up. And we're just going to let those bees soak in Everclear until you collect your fruit set data 2 weeks after the start of the experiment. And so what you're going to do is start the experiment on July 31st by collecting your bee biodiversity data that's contributing to the pollination services over the next 2 weeks. And then at the end of those 2 weeks, you're going to be collecting your fruit set data. So don't harvest any fruit during that period, because we're going to wait to harvest all the fruit at the end of that period, and then we're going to do some measures of that fruit to get an idea the pollination service that's being provided across all of these different farms. So on the left hand side you can just see that there's a mailing box here. I've given you a mailing box, and I'll explain on the next page about what the mailing box is used for, and then also here you can see your leak proof containers with your filters folded up inside of them as examples. Okay, so the last thing you need to do for the project is collect your fruit set data and ship your specimens to determine the pollination services on your farm. So 2 weeks after the start of the experiment on August 14th, you're going to collect all of the fruit set data from the sentinel plants. And so on your data sheet, record the number of sentinel plants that you're collecting the fruit from and the number of fruit that you collect. And then also you'll be recording the total fruit weight and noting the type of scale that you've used. So it doesn't really matter what type of scale you used, but just note the type of scale that you are using. And if you can't record fruit weight at all because you don't have a scale, then just skip that step. Record the number of sentinel plants that you're recording these data from, and then record the number of fruit that's set. After you've recorded that fruit set data, there's space on your sheet to record any additional comments about your farming system that you would like to be included in our analysis. After you've done that, you can just fold up that data sheet and put it in the shipping box that I've provided for you. Then you need to pour off your Everclear from the leak-proof containers. Like I said, we can't ship ethanol or anything flammable through the mail, so pour off the ethanol and give your filters a little squeeze so there's no more ethanol left in them, and then cap them up tightly and place them in the shipping box. So once you have your specimens in the shipping box and also your data sheet, you can apply the business reply label that I've provided for you, and you can tape the box closed, and then you can send the specimens and the data sheet via the U.S. Postal Service. It won't go via UPS or FedEx. That you can just take this to the U.S. Postal Service or put it in your mailbox. They'll pick it up and they'll send it straight to me. And then when I receive these specimens and these data, I will enter the data, and I will also identify all the specimens, all the bee species on your farm, and I will be reporting back to you all the different bee species that we found on your farm. And then also we will be correlating those fruit set data to the measure of flowering plant diversity on your farm, and also be looking at the response of the bee community to the floral diversity on your farming system too. So that's kind of the next slide here that I'm going to talk a little bit about the results of the project. Okay, so the expected results of the project are that we're going to correlate fruit set, pollinator, and floral diversity. All three of those will be correlated. And we expect as crop and bee biodiversity increases, we expect to see increased pollination. So that's what you can see here, is that with low crop diversity, you get low pollination, but with high crop diversity, you get increased pollination, similar to what we see over here in these farming systems. And then also as you see increasing bee biodiversity from one to many species, or increasing evenness, which is the division of, or the abundance of each community member in respect to each other, you see increasing ecosystem services. So we're going to be able to do a lot with these data to figure out what's contributing to pollination services on your farm. So while logical, this has never been shown before, and we believe it would provide farmers with greater incentives to diversify their crop production systems. We also expect greater bee biodiversity as cropping systems diversify, so a lot of niche partitioning to be going on, and this provides incentive to diversify just strictly for conservation purposes. So even if we don't see increases in pollination services, but we see increased diversity in bee species, then even though it might not be providing increased pollination on your farms, it's probably important to conserving bee species in the larger scheme of bee conservation worldwide. In tandem to your experiments, I will also be complementing your studies with cage studies and field research throughout the Puget Sound region. So I will be running similar sentinel plant experiments, which have already begun on 22 farms and urban gardens throughout western Washington. And this will be compared to and enhance the citizen science data. And then in addition to that, I'll be running these cage studies with differing amounts of plant diversity and using a very controlled bee community to look at how strictly the diversification of the farming systems changes pollination services. So rather than just having that correlational data like we'll be collecting on our farms and on your farms, we'll be able to look and see how the causal relationship between plant diversity and pollination occurs. Okay, and that's it. So the conclusion of the course is that pollinator monitoring is hopefully going to contribute to global conservation. So if you have any questions, please let me know through email. And unfortunately, since you folks are not in class, I'm not able to give you the bee station part of the class, but if you ever want to come to another citizen science class, come check out our website and you'll be able to see a lot of these different bee species in person. And I'll give you a tutorial on the different bee species you might be finding in your farming systems. So here's a materials checklist. You also have a materials checklist at the end of your methods document. So be aware that that materials checklist is there, and then also all of this stuff is being provided to you via mail except for the ethanol. And I believe that's it. The ethanol is the only thing that I really cannot ship to you. And then anything on this list that says not provided is something that I cannot ship to you too. And like I said, please contact me with any additional questions or concerns. A few acknowledgments here at the end. Many thanks to the farmers and urban gardeners of Western Washington that contribute to the preliminary data for this project. Many thanks again to our funding sources, my advisor, and research partners. And then, of course, many thanks to Washington State University Extension and also the Oregon Extension Service for helping us reach out to some of the Oregon farmers. And a big thank you, and I look forward to working with you this year. So I appreciate you coming to the tutorial, the online tutorial, and I look forward to working with you this summer. And make sure to review your methods document and all the documents that I'll be providing for you for additional details on how to execute the project. And please contact me with any questions, and you will be receiving your supplies in the mail shortly, including your sentinel plant and all of the other supplies that I detailed in this presentation.